Good evening,
Okay.
everybody from Hyderabad. Welcome to the friends and students of Alliance Française in India, Campus France and Institut Français in India. Science Talk at the Alliance is an initiative of Alliance Française Hyderabad to share the ongoing and long-standing science conversation between India and France. And who better than Dr. Srini Kaveri could highlight the excellency of this scientific dialogue. Dr. Srini Kaveri is going to deal with a very topical issue today. His talk is titled Coronavirus and Immunity, Basic and Therapeutic Aspects. Dr. Srini Kaveri is heading the National Center for Scientific Research, the French National Center for Scientific Research Bureau in India, Embassy of France in New Delhi since 2015. He's Director of Research in INSERM, which is affiliated to Sorbonne University. INSERM is the French National Institute of Health and Medical Research. Dr. Srini Kaveri received his veterinary medicine degree in India and his PhD at the Pasteur Institute in Paris. His postdoctoral research training was in the University of California. He joined the CNRS in 1990. So the research team that he is leading in INSERM works on immunopathology and immunotherapy with a special focus on autoimmune and inflammatory diseases. According to Dr. Kaveri, it is crucial to have a better understanding of the immune response in coronavirus infection. Hence, today he proposes an overview of the immune response in COVID-19, and he proposes to look at the therapeutic and vaccine options. After this short presentation, Ambika will ask few questions. Bonsoir, Srini.
Bonsoir, Samuel. Merci beaucoup.
Merci à vous. I think, Srini, you are talking to us from Paris.
Yes, I'm talking from my home in Paris. And well, if you, may I start now?
Yeah, the screen is yours. Sorry for this small delay, please.
No problem. Thank you very much. And sorry for the little technical snag a few minutes back. Welcome to you all, and good evening for those people there out in India. Before I start, I would like to thank once again Samuel Berthe for this interesting opportunity that he has given us to discuss today about coronavirus and immune response. I also would like to thank Jerome and Ambika in Bangalore and Deepali in New Delhi. Before I start, I would like to thank once again my team in Paris too, because without their efforts, I don't have much to tell you. As you can see, it's a happy little crowd, the team, international team, quite a few countries are represented there. But then this is not surprising. Most of the teams, most of the research teams in France are quite international, actually. And I don't know, those people who have been to Paris would recognize this is Jardin du Luxembourg on a bright spring lunchtime, I guess, picnicking there. If you walk on the right side, about 300 meters, you have Panthéon. And I'm sure you know, some of you know what Panthéon is. And what you see at the far end is the Senate, Le Sénat. And behind that, if you walk down about 200 meters, you have Théâtre de l'Odéon, and go down, you have Boulevard Saint-Michel. You take a right turn, you are in Faculté de Médecine or Cordelier. And that's where sometimes when these people are in good mood, they work. And when they work, they're very efficient. And for reasons that I don't understand, I have a strong affinity, in addition to all those other nationalities, for Indians. Quite a few Indians, actually. On the right-hand side is Vani, who has now come back after her PhD in my group, and she is in IIT Madras. She's an assistant professor. And this shining head here belongs to Jagdish. And Jagdish has been working with me for the last 20 years, and he will be coming over to India and will be heading the Department of Life Sciences in IIT Palakkad. And you have a bunch of other Indian students, and Pushpa Hegde, she is back in Sinjin in Bangalore, and Nimesh Gupta, he is heading a group in National Institute of Immunology in New Delhi. I also work quite a bit with the clinicians, because most of the questions that we ask in the lab have a lot of clinical orientation. So I'm extremely thankful to my clinician collaborators in France, and some of these people are very actively involved in the fight against coronavirus or the COVID-19. And on the right side, you see a bunch of my collaborators. I have not listed all of them. And as you may also see, I have collaboration with IISc Bangalore, Pondicherry, JIPMER, and Bombay, among other places. So before I start, I have a small disclaimer, if you allow me. We are talking to Alliance Française crowd in Hyderabad, and Hyderabad being the biotech I feel everyone is a biotechnologist there. And I even feel a little guilty that what I'm going to discuss today, you already probably know everything. So you should bear with me, okay? And another thing is scientists have this bad habit of using a lot of jargon. And this, if you hear some smutty words or foul words, don't go away. Don't shut your computer and don't go away. They're just immunological terms, okay? So stay with me, please. So let me start. Before I start, I'll share with you a secret and a sacred mantra, okay? And this is very important. You may want to recite that every morning, actually. Whenever you discuss health or the other side of health, which is disease, you must keep in mind these three aspects. The genes, you know that genetic component of every individual is very, very important. And this controls all your body activity called physiological functions. But this, even more important than this, or almost equally important, is your environment. To me, everything outside of your genes is environment. And environment doesn't necessarily mean Jardin du Luxembourg, which is beautiful, bright, and all that. It also means the stress that you would be going through. You are living, the lifestyle, your food habits, your sport or no sport, or drinking or smoking and everything. But environment also involves a lot of microbial, microbes around us. And in fact, bacteria and this microbial world is a very fascinating world. In fact, a whole bunch of bacteria, we have millions of bacteria in us, around us, on us, and many of them are very useful actually. We want those bacteria, and they provide what we call as the normal flora. But then in the environment, there are also some nasty bugs, and those are pathogenic, and we don't want them. And when they get into your body, you have to get rid of them as quickly as possible. So you must have learned in your high school, there is what is called the first line of defense or the front line of defense, which is in immunological terms, it's called innate immune system. Normally, you must have heard the skin, the mucus, the tears, the saliva, the hair, etc. That all protects. This is the first line defense. Most of the times, if the pathogen is in a small level and not very virulent, the first line of defense can work very well and gets over this. But if the pathogen is a nasty one, it comes in a big number and more virulent, then there is the other immune compartment that kicks in. That's called adaptive immune system or adaptive immune compartment. And in this, you have a whole lot of cells and molecules. I'll show a few of them, which are the key or the critical ones. One of those is the phagocyte or dendritic cell, we call them. And these cells, they capture these pathogens and show it to the T cells. These are the T cells that are called T cells because they come from thymus. And the T cells are very important, and they secrete some molecules called cytokines, and cytokines are also very important. Those cytokines provide signal to the sleeping B cell, the B lymphocyte, another group of cells which are sleeping. Now they are kicked in and they start producing antibodies. You must have heard about antibodies. They are represented mostly as a Y-shaped molecule, and they are very specific. Unlike the first line defense or the innate immune system, this immune compartment, the adaptive immune compartment, is very specific. Now, the antibodies will try to identify, and they go hunting all over the body and see where the pathogen or the microbe is. Imagine by then the pathogen, the virus, bacteria, whatever has already reached your lungs, the antibodies go and find them and catch them, capture them, and try to destroy them, and they need some help. So they call in for other cells, the macrophages. Macrophages and neutrophils, they start secreting some obnoxious molecules, dangerous chemicals, biochemical substances, and they try to get rid of the pathogen. Normally it works, but then the T cells are not sitting quiet. They also inject in more harmful substances, harmful for the pathogen. And the T cell compartment is even more interesting. When there is a viral infection or a cancerous cell that's coming in, there is a particular type of cell called the cytotoxic cell. And as the name suggests, it is toxic to the particular cell that has the virus in it. So what does this cytotoxic T cell do? It destroys, it kills the cell that is infected with the virus, and therefore the virus can't survive anymore. It's exposed to the immune system. All this works perfectly well, and you have eliminated your pathogen that's from the system, and you're sitting pretty. And this works to perfection. Everything is controlled, and it works so well that there is no dirt left. All the dirt and everything is cleaned and back to, back on track. But there is something else that happens, and that is those T cells and B cells, they generate one more set of cells called the memory T cell or the memory B cell. As the name indicates, these memorize. What do they memorize? They memorize or remember this particular infection, so that when there is an infection, once again, reinfection, these cells kick in very quickly. They destroy the pathogen very efficiently and very fast. So this concept of memory T cell and B cell is very crucial for vaccines. I'll talk about that a little later. Now, your immune system is very finicky, very tricky. It has to be delicately balanced. You do not want a depression of your immune system. You don't want an exaggerated immune system. You don't want a misdirected immune system. Imagine for a minute that a child is born because of a genetic defect. One of these components is missing. If any one of these components of the immune system is defective or missing because of a genetic defect, that particular child will be immunodeficient. Do you remember the baby bull or the bubble baby? This child on the right side here cannot combat the infections. It has to stay in the bubble, and if it comes out, it cannot even face the challenge of the infection. And therefore, it has to stay in the bubble. And what Professor um, uh, Alain Fischer did in Hôpital Necker in Paris is that he had this ingenious idea of inserting or correcting the defective gene so that this baby can come out. It's a fantastic idea. There are beautiful studies that are going on, and there are some hurdles, but we'll get there. So you don't want to be immunodeficient, and you don't want to have an exaggerated immune response either. You know about allergy. Okay, you are allergic to this substance, that substance, and allergy is not nice either. But then there's something even worse. Imagine for a second that there is no infection, but your immune system is activated. Somehow it is activated, and it is targeting your own tissue, your own pancreas. The same component, the T cells, which are very aggressive, the B cells through antibodies, macrophages, neutrophils, and all those obnoxious material, they come and attack your own pancreas. Then what happens? Your pancreas is destroyed and you can't make insulin anymore, and you have an autoimmune type 1 diabetes. That's not good news. And instead of your pancreas, if it is your joints, you have arthritis. If your nervous system or brain, then you may have multiple sclerosis. Not good news. So immune system has to be well balanced. It has to be working in a perfectly concerted manner. Nothing excess, nothing misdirected. That's the background for you on immune system. Let's go quickly to what happens in coronavirus. If I had shown you this picture before Christmas, you would have said, what is this? But now you have been exposed to different shapes, different colors, different forms, different flavors of this virus. So much so that if I show you this picture today, you may even ask me, who is that guy on the right side? That is the notoriety of coronavirus. But then coronavirus have been there for much, much before the superstar, actually. They have been hanging around. If you see the molecular dating, it seems they were there even some millions of years ago. They were there in bats. They normally stay in bats in this particular family of viruses, but sometimes they have this bad habit of jumping across the species. And when they do that, they can cause some problems. It seems these viruses have jumped from bats to pangolin, and then one such example is the famous SARS-CoV-2, which is causing this pathology called COVID-19. A quick look at the virus. I'm sure you all know, you have seen this picture so many times, I'll not go into the details. it has these spikes on its surface, that corona-like structure of the sun, and that's why it's called coronavirus. And you have the genetic material, which is RNA. I'm sure you have heard of DNA, RNA. This virus is an RNA virus, and that's its genetic component, and that can control quite a few its activities. So what does it do? It infects you. You have seen this picture so many times over the last 3 months. Whenever there is an aerosol or droplets, if you inhale or if it goes through your eyes or your mouth, it goes into your digestive system, causes some digestive disorders. But if it goes to the lungs or your respiratory tract, what happens? Let's see. This rectangle here represents one cell on your lung lining. Okay, lungs have this epithelial cell, and this is one cell. On the surface of cells, there are some molecules. They're called receptors. Don't worry about the name. These spikes, the famous spikes, they sit on that particular receptor, and that's a portal of entry for the virus. It gets in. Once it gets in the cell, it completely hijacks the host cell. It uses the entire machinery that the host cell provides for its own benefit. Benefit of what? To replicate, to multiply. It multiplies very fast. Imagine you had a dinner with a friend of yours, and that person had a virus, I mean, the coronavirus. From last night to this morning, in 10 hours, this virus has multiplied more than 1,000 times, which means in your cell, you have 1,000 copies from one cell, and your body has millions of cells. So you have several millions of viruses the next morning. The whole cell is bursting with viruses, and the virus jumps from one cell to the other and starts infecting other cells. And the whole thing gets rampant, and you are full of viruses. But then the immune system is there. It kicks in. It doesn't like this. It sees there's a danger. We should do something about it. So what does it do? You know that. Now the coronavirus has entered in. The immune system recognizes the first line defense doesn't work now. The adaptive immune compartment has come in, and these cells, specialized cells called dendritic cells, they capture the virus, shows it to the T cells, and these T cells differentiate into other type of cells and cytotoxic T cells. And as you know, they start secreting inflammatory cytokines, and then the cytokines also provide signal to the B lymphocytes. They make antibodies. The antibodies recognize the virus. They capture them, and they want to destroy now. The virus has to be destroyed, has to be eliminated. It calls in the help of macrophages, the neutrophils. They start secreting all possible substances like reactive oxygen species, as I told you earlier, and many other substances. And this works to perfection. The virus starts getting eliminated one by one. The virus goes. You're back on track. But then there's something else. You have a bonus. You even have these two bunch of cells, you know, the memory T cells and memory B cells I was talking to you about, which means that in case you get the reinfection in a few months, hopefully not, these memory T cells and B cells are ready there, and they can come in anytime and then ensure that you don't get the infection or a full-blooded infection, because they can kick in very quickly and get rid of the pathogen through other help, the T cells and B cells. Now, then why are we making so much fuss? 60 to 70% of us come out unscathed, you know, and many of you, maybe even sitting there, you might have already gotten over that without even realizing. You had a small cough, a little fever, and then 3-4 days you're walking upright. Then why the fuss? Why are we making such a panic about it? The fuss or the panic is because this virus is new. This virus is very different, unlike other viruses. This is extremely contagious, and it can spread very fast, and it is extremely dangerous for the time being, because we know its infectivity is much more. And there's one more thing. See, most of those 60 to 70% of the people are having a solid immune system. They're robust, they're young, energetic, doing all sports and all that. They can ward off the infection, as I showed you a few seconds ago. But then there are these unfortunate individuals, elderly individuals, diabetics, the obese individuals, or cardiovascular disorders, people with those diseases, or other comorbidities, meaning other disorders. Their immune system is fatigued. They don't work the same way. Their immune system cannot cope up the same way as a healthy individual. So what happens in these cases? In these cases, the immune system is confronted with the virus, but these are unfortunate individuals, and they try to The immune system tries to fight the infection. It calls in the T cell compartment, the B cell compartment, the whole tralala. You already know that now, you are specialists. And the macrophages, the neutrophils, everything, everybody comes in and tries to ward off the infection, but the infection is not going because the immune system is not working optimally. More virus comes in and more substances are released. These are obnoxious substances, you know. In a good controlled immune system, all is well. These substances come and go, do their job. But now it's not going because there's more infection coming in, more obnoxious material coming in. You are in a bad shape. The whole system is in complete panic, and there is a destruction beginning. The whole immune system is on fire practically, and that's why we call it inflammation. Inflammation is flamed up, you see. This is not good news. What happens at that time is your immune system is so tired and exhausted that it goes a kind of eclipsed, and that's taken over by this storm called cytokine storm. As I told you, these bad substances, cytokines, which were so good when we needed them, when they're controlled. But now there's no control. Uncontrolled immune response means severe inflammation. And then you have so many other associated problems like clotting disorders. You have micro clots all over the body. They start clogging. Lungs get injured. They start fibrosis, and they're not working well. The cells start dying. Immune system is exhausted, paralyzed. A shock is set in, and multiple organ failure, and that's not good news. And you have switched on the panic button, self-destruction, and some of these individuals unfortunately could not survive. So what are we going to do about it? Let's talk for the next few minutes about currently what approaches we have for treatment. Actually, there's nothing. No, I'm exaggerating. There is no specific anti-coronavirus drug yet, but the knowledge from other viruses has taught us that there are so many drugs that are possible that we can use. And there are many other approaches to control the associated pathology. Plus we have the vaccination. Not yet, we will have that. Now, one or two words about the antiviral drugs. Now, we have learned from other viral infections and this coronavirus infection that viral entry is there, and we can stop the entry of the virus. And we know that virus gets in and uses the machinery of the cell. So each and every step can be blocked. And for each and every step, either the membrane fusion or for assembly of the different parts of the virus for the replication, there is a way of blocking. And this is the knowledge that we have gained from many other viral diseases. And you must have heard about chloroquine and how its efficacy. And India is a big manufacturer of chloroquine, and this has made a lot of news. And several trials are going on with chloroquine too. I'll not go into the details of more about the antiviral drugs, but we will discover many more antiviral drugs. But then you need other approaches because, as I told you, the lung tissue is damaged. We cannot breathe anymore. The oxygen is not assimilated. So you need oxygen therapy or mechanical ventilation. And I also told you that the immune system is gone crazy, and it's exaggerated activity, and therefore you have to suppress that immune system. So at one point you can use the corticosteroids, but you should be careful when we use that. And as I told you, there is excessive inflammation. So immunotherapy is also being practiced, particularly some cytokines which are overactive like IL-6. There is a therapy to stop that using monoclonal antibodies and polyclonal antibodies. And in fact, there are some studies with the BCG, the Calmette-Guerin vaccine for tuberculosis, and that is also tried. And convalescent plasma. Now you know that when you recover from this coronavirus, your plasma will have antibodies. So the plasma is going to be useful because there are antibodies. How to use this plasma as a source of antibodies against specific, now these are specific against corona, and there are studies going on. And just this morning, I saw a tweet from Shekhar Mande of CSIR, and CSIR has, is going to work with Ayush India on traditional medicine. And actually, 4 compounds, different combinations are being used to see the efficacy of that. I'll not go into the details of this. A few words on vaccination. Vaccination is important. And the more number of people are vaccinated, hopefully soon, you will have a herd immunity. And when 60 to 70%, luckily, hopefully, are vaccinated, you establish a kind of a herd immunity, and that is very important, and the virus cannot propagate much. But then to get there, you have a lot of work. The vaccine has to be safe, it has to be efficient, and one single shot should give you a long-lasting memory. So this is going to take some time, but there are a lot of efforts, because we know each and every bit of this vaccine, I mean, this virus now. So currently there are approximately 120 different vaccine candidates are being studied. Government agencies, the companies, academic labs, all of them are putting a lot of effort on trying to get a new vaccine against coronavirus. 8 of them, these vaccines have already reached clinical trial stage. And in India, a lot of effort is being made by ICMR, CSIR, DST, DBT, and institute like Serum Institute of India has a huge potential. It's one of the biggest institutes for manufacturing large quantities of vaccine. And so all these people are making efforts. And on the French side, yes, this is the country of Louis Pasteur and Françoise Barré-Sinoussi. You know, Françoise Barré-Sinoussi discovered the HIV or AIDS virus. So they know one or two things about viruses. Many agencies like INSERM, CNRS, ANR, they are all putting in efforts. And there's a very interesting consortium called REACTing, which has established a European level consortium doing clinical trials. And Institut Pasteur is putting in a lot of effort. I'll not go into the details of that. And the idea is to come up with appropriate diagnostic tools or vaccine and plasma therapy. And Pasteur Institute is putting in a lot of effort. And several French companies like Sanofi are also putting in efforts in international collaboration and to come up with a vaccine. Now, I'm a born optimist. And the road to recovery for coronavirus would be, I hope that will be soon. More than 1 million people around the world have already recovered from this virus. Mathematical models estimate that between 99 to 99.5% of the people who would get this will also recover. And there will be new and specific antiviral drugs available soon. The vaccine trials will also happen. And in my opinion, coronavirus can be controlled and will be controlled. And hum honge kamyab, will be successful. I'd like to thank you all for your attention, and I'm ready for questions.
Thank you so much, Dr. Srini Kaveri, for this wonderful presentation and for making really intelligible the complexity of our immune system, and also for opening up perspectives on the upcoming discovery of different cures to the current pandemic. Now, I would like to introduce Ambika, who's going to ask a few questions about the talk that you just gave. Ambika is PhD in cell biology from the National Center for Biological Sciences in Bangalore, which is currently functioning as a national COVID research center. And she joined in 2018 the Embassy of France in India as the scientific coordinator for the South Zone, aiding, helping in industry and academic cooperation. Ambika, can you hear me? Are you?
Yes.
Yes.
Yes, very well.
Good evening, Ambika.
Good evening, Samuel.
You are talking from Bangalore, I suppose?
Yes, from my home in Bangalore. Because of the lockdown situation. And thank you, Samuel, for the introduction and for having me here. And thanks to Alliance Française Hyderabad. And Srini, I must say it was a fantastic talk and very insightful and relevant to our current situation. So I know that a lot of questions have been already answered through your wonderful talk, but I would like to share a few questions to highlight some perspectives even more with much more clarity. So one of the questions I had after listening to your talk is that you mentioned that the virus enters into our body and triggers immune response, and which could result in self-destruction of our own cells. Now, when you inject vaccine into our body, you mentioned that that also will result in triggering our immune system. So how can one ensure that the vaccine will not have the self-destructive consequences like the virus?
Yeah, thank you, Ambika. That's a very interesting question, because in fact, what is vaccine or what is vaccination? Vaccination is, in my opinion, it's actually cheating the immune system. And what we try to do here is to imitate an infection, but not the real infection. So how do we ensure that? I mean, I have one or two pictures for that. There are various strategies. You actually take a virus, make it inactivated. There are various ways of inactivating it, ensure in several preclinical studies that it does not induce any, there is no capacity, it has lost completely the potential of inducing any disease, and you inject that. Or even better is you take only a part of the virus, okay, which is important triggering the immune response, and you insert in another vector, completely denuded. For example, you can take adenovirus or measles virus, strip it off of all the potential to induce the disease, but you you introduce only some important parts, like this spike here, the spike that are on the coronavirus, or you take only the spike protein. You can actually prepare the spike protein outside the body, and you can use only that, which means you don't have the virus now. You only have the spike protein, and it cannot cause a disease because there's no virus here. Or even better, a part of the DNA, actually, you can do a genetic engineering method wherein you take a piece of DNA and make it produce a particular protein of the virus, like a spike protein, others. Okay, so there is the DNA vaccines are also possible, and then there is even RNA vaccine, and this is also going on. The studies are going on. So, in brief, none of these has the potential to actually induce a disease, because there is no potential to induce any of the pathology. On the other hand, when you inject them, there is the component, the immunological component, antigenic component, which can trigger the immune system do the same thing as cytotoxic T cells, as I told you, or the antibodies that are able to neutralize. But more interestingly, these vaccines, they also have the ability to induce the memory B cells and memory T cells. And this is the critical thing, which means all these vaccine candidates should be able to induce memory B cells or T cells, so that when there is a reinfection, these memory B cells and T cells can take care of. Okay, this is how we ensure a vaccine works.
Yeah. Okay. Thank you, Srini, for perfectly answering the question and showing that science is no magic. And so this leads to my second question about vaccine again. So you mentioned that if we take the vaccine and assume that the vaccine is currently available in the market, and we can fight this particular virus with the help of this vaccine. But recently, there has been a lot of news floating around the fact that there is another mutant version of this virus which is available. So, I mean, which is emerging. So, do you think the same vaccine can do its action in both these conditions? And if yes, how long can our immune system, remember this and keep the action alive?
That's a good question. Yeah, I know that there's a lot of news, or how do you say, noise going on about mutation and what happens if there is a new mutant that comes out, and if that is able to, whether that will cause one more pandemic. Now, before I answer, what is a mutation? Let's go back to that particular image that I showed you. The virus enters into the body and it replicates several thousand folds. Okay, I told you overnight it can make 1,000 copies, but imagine this machinery is not that foolproof. Small variations are going to happen when you are replicating thousands and millions of times. There are bound to be small changes, and each of them is a small mutant, and these are not going to cause, every mutant is not going to cause a pandemic. And what we know about coronavirus more until now, the studies show that there is a stability, and there are small mutations. I mean, in fact, there's a Scottish paper that has come out very recently where they have identified more than 7,000, changes, or the mutants, if you want. Okay, the small mutations, which are not necessarily emerging as a new pathogen or a new pandemic. So, answer to your question, these mutations, all these mutations do not necessarily mean new pandemic or a new, pathogen. So far, if our knowledge goes well, correct, I mean, if I understand correctly, the vaccines that we have designed or we are going to design should be able to impart immunity and protection for quite some time. Although studies have to be done to say how long will be the protection. The time will tell us.
Okay, but there's a lot of hope.
Yeah,
so
yes.
I think we are going to close the session soon, but before that, I have a final question to you, which is a bit personal and professional at the same time, because you've been in the field of immunology 30 years or so. So while we are all together in dealing with this crisis, what is the role of your laboratory and you in dealing with this pandemic?
Thank you. My lab has been working for last 30 odd years on one particular area, which is called immunotherapy, and in particular on antibodies. You remember those Y-shaped molecules that I mentioned to you about, and we know about the existence of antibodies since 120 years, ever since they were discovered by Kitasato and Behring 120 years ago in Germany. But then we still do not know much about these antibodies. There's a lot to understand about the function of these antibodies. And my lab has been working on antibodies, particularly antibodies in health, in physiology, and antibodies in disease. But more importantly, how to exploit these antibodies as immunotherapeutics. In fact, in COVID context, what we are trying to do is the pooled antibodies from several thousand donors, whether they are able to prevent the entry of virus into the cell, and more importantly, whether we can use these antibodies as immunotherapeutics because they can subdue or control the inflammation. And inflammation, as I told you, is an important component of COVID-19 disease. So we are hopeful that a immunotherapeutic approach to control the inflammation would be in one way that we can contribute to the understanding or our contribution in this area.
Well, thank you so much, Dr. Srini, Kaveri, and Dr. Ambika, for your very, very kind and very insightful participation to the Science Talk at the Alliance. Well, the research is on, and I think there is, as you mentioned, hope to find various ways of confronting the pandemic. I mean, as you really have clearly exposed, there are different direction in which the research is operating right now to confront the disease. So thank you very much. I'd like also to thank the Alliance Française in India, the Institut Français in India as well, and Campus France. A special thanks to Jérôme Bove and to Isabelle Jetli. These talk was also streamed in Bangladesh, in Alliance Française Chittagong. It will be uploaded on our Facebook page and on Institut Français YouTube channel. Thanks a lot. The next Science Talk at the Alliance. Thank you, Srini. And what a beautiful flat, I must say.
Thank you very much. Yes.
It seems to be a bit chill in Paris, but
it is chilly. Yes.
But the light is
it's a beautiful sunny out there. Yes.
Thank you. Thank you, Srini. Yes. Thank you, Ambika. The next Science Talk at the Alliance will be next Thursday, this time at 7 o'clock dot. And Dr. Anne Chapuis will share her work on visual data analytics about the coronavirus in India. The former Chief Secretary of Andhra Pradesh, Mr. Tucker will be the discussant. And we will also have very soon another Science Talk at the Alliance on a totally different topic dealing with women and sports in India. Thank you again, once again. Bonsoir, Srini.
Bonsoir, merci.
Bonsoir,
Bonsoir.
Ambika.
À
Et
bientôt.
à bientôt. Au revoir.
Au revoir.